The idea with basement membrane treatment is that we target the primary gene deficiency, that is loss of laminin alpha-2 chain. There is also a lot of work done modulating the cellular events caused by loss of laminin alpha-2 chain. And in that sense, we're targeting the secondary disease driver. So I will talk a little bit about that too. Then the third speaker, that is Durbeej, maybe you can just wave your hand so that everybody sees you. She will talk about CRISPR-Cas9 as a way to genetically correct mutations in MDC1A. And then there's a fourth group of strategies, so to say, and that is cell therapy. And Professor Cousseau will talk about that. He's the third speaker in this session. But actually, more than 50 different therapeutic approaches have been evaluated in MDC1A mouse models. Some with good results and some with less good results. And of course, I only have 30 minutes now, so I really apologize for not being able to discuss all of these today. But basically what I will do now during this lecture is that I will give you a short introduction to laminins, because these are, the main molecules that we are discussing today, and I will tell you a little bit about basement membranes. And then I will give you a few examples of basement membrane treatment. And I will also give you some examples of how to modulate cellular events that are caused then by loss of laminin alpha-2 chain. And then I will end with giving you some examples of combinatorial treatment. And all of this has been done in animals, I should say. So the molecule of interest here is these cross-shaped molecules. They are called laminins. So they are composed of 3 different chains, an alpha chain, a beta chain, and a gamma chain. And laminin 211, that's the laminin isoform that is highly expressed in muscle and nerve. And MDC1A is then caused by mutations in the alpha-2 chain. Now there are many different laminin isoforms in our body, so I will also be talking about laminin 111 today, that's this molecule here. So the main difference here is that here is another alpha chain, this is the alpha-1 chain. And this laminin isoform, laminin 111, that's, for example, expressed in our kidneys. And these laminin molecules, they are found in structures that are called basement membranes. So basically, basement membranes, they are these thin sheets of protein structures that are found outside the muscle cells. So they basically surround muscle cells. They're also present in peripheral nerve, where they surround Schwann cells, and these Schwann cells are the cells that make the myelin surrounding the axon. So this whole package, so to say, is surrounded by a basement membrane. And also in the kidney, there are basement membranes. There we have cells that are called epithelial cells. They sit here on the basement membrane. So this basement membrane is rich in laminin 211, whereas this basement membrane is rich in the other laminin isoform, laminin 111. And these laminins, they form networks. So these cross-shaped molecules, they spontaneously develop into networks. And this is a process that is called laminin polymerization. And Peter Yushchenko, maybe you can wave your hand there, he has done seminal work in increasing our understanding in how laminins polymerize. And we will come back to laminin polymerization a bit later. And so these laminin networks, here is now shown only a single laminin molecule, they are attached to the cells, for example, a muscle cells, via interactions of cell surface receptors. So here you're basically looking at the cell, here you are looking at the basement membrane. So at the cell surface, there are proteins called cell surface receptors, and laminins, they bind to these cell surface receptors. So 2 well-known cell surface receptors, they are integrin, you cannot really see here, it's called integrin, and the other one is dystroglycan. And laminins, they bind tightly to these receptors. And thereby, by this interaction, they form a strong structural support, but laminins, they also send signals into the cell, telling the cell what to do, for example. So in MDC1A then, what happens is that laminin 211 is missing because there's mutations in the alpha-2 chain. That in turn leads to a secondary reduction of one of the receptors, the integrin receptors, and in muscle there's a integrin receptor called integrin alpha-7 beta-1, and it's the alpha-7 subunit that is secondary reduced. And then what happens is that there is a compensatory upregulation of yet another laminin molecule, and here you see this laminin molecule is T-shaped rather than cross-shaped. And this laminin molecule is called 411. So this molecule, it cannot polymerize because it is lacking the most end part or end-terminal domain that is important for laminin polymerization. And this molecule cannot bind well to the receptors either. So basically this compensatory upregulated laminin is a non-functional laminin, so to say, if we say it in a simple way. And this, of course, then leads to MDC1A. So what happens in MDC1A is that these structures, these basement membranes, they basically break. So there's a broken basement membrane around the muscle cell, and there's a broken basement membrane also in the peripheral nervous system. But here, the kidney basement membrane, that stays intact because there you have another laminin 111. So loss of laminin alpha-2 chain, it further leads to a disrupted basement membrane. And then muscle cells, we can say that they break, they degenerate, but they also have the capacity to be repaired. So they have a little bit of regeneration capacity. They can sort of repair themselves initially. Another typical hallmark of laminin alpha-2 chain deficiency is that there is increased muscle cell death. And that's a phenomenon that we know as apoptosis, so I will tell you a bit more about that later as well. Another typical hallmark, what you see in a muscle biopsy, is that there is a lot of inflammation going on. We see a massive inflammation at certain stages. And then there are also increased muscle fibrosis. That's we can say a repair system as well to some extent. So there's massive, this disease is characterized by massive inflammation and also massive fibrosis. And what we have shown also that the metabolism, muscle cell metabolism, is also impaired. So now, if we talk about the therapeutic strategies for MDC1A, and we talk about the first group, that is basement membrane treatment, how can we sort of restore the basement membrane? How can we treat the basement membrane? And Eva Engvall, more than 20 years ago now, she showed in animal models that it's actually possible to put back the missing laminin chain. So in animal models, she put back laminin alpha-2 chain, and she could sort of rescue the phenotype in an animal model. In my lab, we have been thinking about alternative strategies. We want to use another laminin, for example, laminin alpha-1 chain instead, mainly to avoid unwanted immune responses. So I'm going to tell you a bit about this. Work from Marcus Rugg lab, maybe you can wave your hand as well, here's Marcus, and together with Peter Yushchenko, they have developed another strategy to treat the basement membrane. So they are using linker molecules. So I'm going to talk about that too and give a short summary of those two. And Dean Birkin, he's not here today, he has also shown that one way of treatment is actually to restore the integrin, that was the cell surface receptor that was missing. If that expression can be restored, then MDC1A can also be reduced or improved in animal models. So we do all our work in animal models. So these are pictures of the different animal models that are available. So we mainly work with 3 different models. This is the mouse that is called the DY2J mouse. This is the mouse that is called DYW, and here's the DY3K mouse. And these models very well mirror the pathology. So they are very, very good models to study this disease. So the DY2J mouse, that has a milder form of muscular dystrophy, but instead it also develops a very severe hind leg paralysis. So they have hind leg lameness, so to say. But these mice, they have extended survival. They survive more than 6 months. The DY3K mouse is the most severely affected mouse. This mouse, as you can see here, has a very short lifespan. The median survival in our colony is about 3 weeks. And the DYW, it's another mouse model. They also develop a very severe muscular dystrophy, but they live slightly longer compared to the DY3 animals. They live around 5 to 12 weeks. So we were thinking, would it be possible then to correct the basement membrane using this other laminin chain that is present in the kidney basement membrane? So we were thinking maybe laminin alpha 1 chain can replace laminin alpha 2 chain in muscle and nerve. So basically we were asking this question, can laminin alpha 1 chain functionally replace laminin alpha 2 chain in dystrophic skeletal muscle? And we chose laminin alpha 1 chain because that is the laminin molecule that is most similar to laminin alpha 2 chain. It binds to the same receptors. So this laminin 111, it binds to dystroglycan. This is the cell surface receptor. It also binds to integrin alpha 7 with similar high affinity as laminin alpha 2 chain. And then we were asking, can MDC1A be prevented in animals? So what we did, we generated mice that were deficient, they were lacking laminin alpha 2 chain. Instead, they were overexpressing laminin alpha 1 chain. So this is the DY3K mouse that is not treated, so to say, and this mouse instead, it's missing alpha 2 chain, but instead it has laminin alpha 1 chain. So already at 5 weeks of age, these mice are much bigger, and we see that also in older animals. And I have a video here of 2-year-old animals. So remember, the sick DY3K mice, they die at 3 weeks of age. But when you overexpress laminin alpha 1 chain, they live 75% of the mice, they live up to 2 years of age. And it's this mouse that you should look at. This is overexpressing laminin alpha 1 chain. So you can see they are able to move, they don't have any hand-leg paralysis, they're able to stand up, so you cannot really tell it apart from the wild-type mouse, so to say. So we have also overexpressed laminin alpha 1 chain in the other mouse model, that is the DY2J mouse models, that has a less severe muscular dystrophy, but it has a severe hind leg lameness. You can see that here, this mouse, is dragging its legs. This other mouse, so this mouse is the DY2J untreated, this mouse is expressing high levels of laminin alpha 1 chain. So you can see the difference here. This other mouse is dragging its back legs. But then this mouse is moving very freely and can easily stand up on its back legs. I have yet another video. So here we place mice on a rotating rod at different speeds. So in that way we are measuring the neurological function. So this mouse to the right here, that's a DY2J mouse, and you're going to see that this mouse cannot stay on the rotating rod, not even at low speeds, because it's falling off. This mouse here is expressing laminin alpha 1 chain. You can see eventually he falls off, and this is a very low speed. And here again, here is another mouse overexpressing laminin alpha 1 chain, and this is a wild type littermate. So here we're rotating the rod at a higher speed. And this mouse can stay on as good as a wild type animal. So basically, this is all work done by Kinga Gavlik in my lab. So what she has shown during the last, it's almost 20 years now, she has shown that laminin alpha 1 chain significantly reduces muscular dystrophy. It also reduces the peripheral neuropathy, and what I didn't show you, it also reduces cardiomyopathy in MDC1A mice. So the question is if laminin alpha 1 chain overexpression can be a future therapy for MDC1A patients. And if so, how can we deliver it to the patients? That's a major hurdle. How can we go from mice to humans? And the question is, can we use viral vectors? And probably not at the moment, because very few of the viral vectors can accommodate large genes such as laminins. They are extremely large. So, at the moment there are no good viral vectors. But protein therapy, for example, could be an approach, and I will show you that soon, and also endogenous upregulation. So Dean Birkin, I told you about his work on when it comes to integrin alpha-7, he showed in 2012 actually that if you inject the laminin 111 as a protein, so that's direct injection of the protein, you can actually prevent muscle disease in the other mouse models, the DYW mice. So here you're looking at the cross-section, that's a TA muscle here, and you can see that it's quite dystrophic. And then you inject laminin 111 into the TA muscle, and you can see that muscle morphology looks much improved. Here you're looking, here they did an IP injection, here you're looking at the triceps muscle, and you can see that it's quite dystrophic. But upon injection of this protein, muscle morphology is significantly improved. And Protheria, that's a company, and it's represented here by Brad Hodges. They are developing recombinant human laminin 111 as an intravenous protein replacement. So he's here if you want to talk to him later today. And then Dri, the next speaker, she has found a good way how we can upregulate the laminin alpha-1 chain using very novel and sophisticated technology that is called CRISPR-Cas activation system. I think you will hear about this in the next talk. So that was our approach. Now Marcus Rigg and Peter Yushchenko, they have found another very clever and innovative way of correcting the basement membrane, and they use so-called linker molecules. And these linker molecules, they are called mini-agrin and alpha-ln ND. And these linker molecules, they have been designed to restore the structure and function of muscle fibers lacking laminin alpha-2 chain. And basically, if we go back to how does it look like now in MDC1A, remember laminin alpha-2 chain, laminin 211 is missing, there is a secondary reduction of the integrin subunit and a compensatory upregulation of laminin 411. Now this laminin cannot bind well to the receptor dystroglycan and it cannot polymerize. But then Marcus, he designed a molecule that is called mini-agrin. And this molecule, mini-agrin, it basically restores the laminin binding to the receptor. And when they overexpress mini-agrin in mice, they are using the DYW mice, muscular dystrophy is very much reduced. So here you are looking at the sick DYW mouse, and this mouse is overexpressing mini-agrin. And these mice, they walk better. You can see that on these graphs, they weigh more than sick animals, and also more importantly, they survive longer than sick animals. So the question is, can mini-agrin be a future therapy for MDC1A patients? And if so, how to deliver mini-agrin to patients? Well, mini-agrin, that's a smaller molecule, so that can more easily fit in a virus vector. And it has been shown that was work from Zhao Xiao's lab in collaboration with Marcus. They showed that adeno-associated virus-mediated mini-agrin delivery significantly reduces muscular dystrophy in DYW mice. And also Stefano Previtali, where are you here? Yeah, maybe you can wave your hand as well. He's there. He has shown that you can deliver mini-agrin with a stem cell that is called mesangioblast, and that can also reduce muscular dystrophy in DYW animals. And then we go to this other linker molecule, alpha-ln nitrogen. And this is what it looks like in the DY2J animals. Remember, these are the animals that are the least severely affected. They have a defect in laminin polymerization because they have a mutation in the domain that is involved in laminin polymerization. And there are also patients that have similar mutations. So in these mice, in the DY2J mice, laminins cannot polymerize because they're lacking this crucial domain. So what Peter did together with Marcus, they designed a linker molecule that is called alpha-ln nitrogen domain. And this is a very simplified schematics of how this linker molecules binds and looks. But the idea is basically to bring back a polymerization domain so that laminins again can polymerize. So by doing so, adding these linker molecules, lamins again can polymerize. So they have overexpressed these linker molecules in the DY2J mice with good results. So here you are looking at the cross-section of a dystrophic muscle, and when this linker molecule is added transgenically, muscle morphology is very much reduced. Here you are looking at fibrosis. You see fibrotic tissue here in red that is also significantly reduced in transgenic mice overexpressing this linker molecule. And so then Marcus and Peter, they combine these two linker molecules. So in DYW mice, they can transgenically add both mini-agrin and this other linker molecule, restoring also polymerization. So basically they are developing transgenic mice overexpressing both of these linker molecules. And these mice, they show remarkable results actually. So transgenic expression of both of these linker molecules very much reduce muscular dystrophy in DYW mice. So here you're looking at the DYW, it's a sick mouse. Here you're looking at the control normal mouse. And here you are looking at the double transgenic mice. So they have very good improved body weight, as you can see here. And here they also live much longer. So they have an extended lifespan up to also more than 2 years of age. So that is very impressive data. And Marcus Rück together with Santera, they now collaborate to develop new gene therapy for MDC1A. And I think Marcus and Peter will be available then at the roundtable discussions if you want to discuss this therapeutic strategy. So that was the work that I chose to focus on when it comes to curing the primary defect of MDC1A. But a lot of work has also been done in modulating cellular events caused by loss of laminin alpha-2 chain. And of course, these approaches, they don't really target the primary deficiency. But nevertheless, we're still, in preclinical phase with the strategies that target primary deficiencies. And meanwhile, it could be also good to look into alternative options as treatment. And so MDC1A is characterized by increased cell death, so muscle cells die via a phenomenon that is called apoptosis. And Reagan talked briefly about that too. And there is also enhanced muscle fibrosis that I will show you, and also I will say a few words about the impaired muscle cell metabolism. But basically what we see this muscle cell death, known as apoptosis, we see it very early in our animal models. For example, so here you are looking at the cross-section of a 1-day-old skeletal muscle, and these red fibers that you can see, they denote apoptotic fibers. So apoptosis is present very early in MDC1A, both in mice and also in patients. And Sweta, there is Sweta, she performed seminal work showing that apoptosis is a disease driver in MDC1A. So what she showed in 2004 was that when apoptosis was inhibited in DYW mouse, then muscular dystrophy was significantly reduced. So here you are looking at the DYW mouse, it's sick, here is a normal mouse, and here apoptosis was inhibited. And basically you can see that this mouse is much bigger. We can see that it's has increased body weights, and they also live longer. So clearly apoptosis is a disease-driving factor in this in MDC1A. And Reagan, she talked about omega pill in a clinical trial, and there is this drug that is called omega pill, and it was originally developed by Novartis. And two different studies, one from Marcus's lab and another study from the state, has shown that treatment with this drug omega pill has beneficial effects in 2 mouse models, in the DYW mouse and also in the DY2J mouse. So when you treat mice with this drug, cell death is inhibited, it reduces weight loss, it reduces fibrosis, and it improves muscle, improves motor function, and also increases lifespan. And this is just to summarize basically a short slide of Reagan's work, that the first clinical trial with omega pill in patients have been performed. This study is called Callisto, and it basically showed that omega pill is safe and well tolerated. So another disease driver in MDC1A is the enhanced fibrosis. And what is this fibrosis? Well, it's the formation of excess fibrous connective tissue. And basically you can see it here. This is a picture from Marcus's lab. So here you are looking at the cross-section of a muscle from DYW mice. And basically what you see here in blue, it's the fibrosis. So basically it's a replacement of muscle with fibrotic tissue. That's a hallmark. And that is actually what makes the muscle weak. The infiltration of this fibrotic tissue. Here you're looking at the cross-section of a normal muscle. So then, of course, anti-fibrotic therapy would be, we're thinking that this could maybe be beneficial in mice. And there is a drug that is called losartan. And losartan is an improved anti-hypertensive medication that is actually emerging as an anti-fibrotic therapy. So several different research groups have shown that losartan has beneficial effects in animals. So this is a study from Joram Nevo's lab in Israel. He's not here today. But here you can see in the DY2J mice, you see now here fibrosis is shown in red. There's increased fibrosis in DY2J animals. But then when you treat these animals with losartan, there is reduced fibrosis. And Sweta has also performed similar studies. So here is a cross-section of a DYW mouse. You can see that this is also highly fibrotic. And when she gives the mice losartan, fibrosis is very much reduced. And also Marcus has used a losartan derivative. So here again is the cross-section from the DYW mouse. You see massive fibrosis, which is very much reduced when you add this losartan derivative. And so lastly, another secondary disease driver is mitochondrial function. So we have shown, I'm going to show this very quickly, that we think that there is problems with the mitochondria in MDC1A. Mitochondria, they can be regarded as the energy-producing factors in the muscle cells. They are present in all muscle cells. But we find that mitochondria are somehow defective in MDC1A. So we think that there is a relationship between laminin, that is outside the muscle cells, and there's a relationship with the outside of the muscle and the mitochondria that are present inside the muscle cell. So therefore we're thinking that maybe skeletal muscle metabolism could be a pharmacological target for MDC1A. So we have tested, these are very preliminary results, but we have tested two different compounds, metformin and NAC. They are called. Metformin is an approved anti-diabetic medicine, and NAC is an approved antioxidant. So we have treated DY2J mice with these compounds separately. So basically here we're analyzing grip strength as an outcome measure. So grip strength is very much reduced in the DY2J animals, but if we give them metformin, the grip strength is increased. They become stronger. Here is again the same graph showing that in DY2J animals the grip strength is reduced, but if you give the mice this other drug, this antioxidant, mice become stronger, and we measure here the forelimb grip strength. And lastly, I just also want to mention combinatorial treatment, because when laminin alpha-2 chain is missing, there is a very complex pathology. And that could actually be the result of a dysregulation of multiple cellular processes. And maybe strategies that target these different processes at the same time could have better effect than targeting only one cellular defect, so to say. And Sweta has done a lot of work in trying different combinatorial treatments in animals. So she has, for example, shown that a growth hormone and an apoptosis inhibitor successfully reduces muscular dystrophy in DYW animals. She has also shown that growth hormone together with this antihypertensive drug losartan, the two of them also significantly reduce muscular dystrophy in animals. And also Marcus Durbeej has tested apoptosis inhibition aminoagrin and showed that they have also additive benefits in DYW animals. So basically, if we summarize what I have said and talked about here today, I have shown you that lambda alpha 1 chain and also linker molecules, respectively, they significantly reduce MDC1A in the different mouse models. I also shown you that compounds targeting apoptosis, one such compound then is then omega-P, a compound targeting fibrosis, for example losartan, and compounds targeting metabolism, metformin and NAC, they have good effects in MDC1A mouse models. And I've also shown you briefly that also combination therapy has been evaluated in MDC1A animals. And of course, this looks good in the animals, but there is still a lot of work to be done. And I have just listed a few discussion points that maybe we can discuss later, but I think it would be very important to validate the different approaches. It would be good to validate them in different mouse models. We now have 3 different animal models, and we should also validate work from each other's labs, so to say. And of course, what we know little about in the animal models is also when is the disease starting. So we need to know when to intervene. And something that we haven't discussed so far, but laminin alpha 2 chain is not only expressed in muscle and nerve, it's also highly expressed in the brain and in many other tissues. So one question that we have not really addressed is, apart from CNS involvement, central nervous system involvement, is non-muscle manifestation. For example, laminin alpha 2 chain is also expressed in the kidneys, expressed in the gastrointestinal system, is also expressed in the lung. So the question is whether there are also non-muscle manifestations. And I think what is great when we meet together like this, that maybe one goal should be to assemble a priority list of approaches to move forward. And such discussions should be held together by patient organizations, clinicians and researchers. And that's basically why we're here today. So thank you.